The ZUMA-12 is a phase 2 multicenter study evaluating axi-cel CD19 CAR T-cell therapy as part of first-line therapy for patients with high-risk large B-cell lymphoma. So previously, axi-cel has been approved for patients with large B-cell lymphoma after 2 or more lines of systemic therapy. This is the first study evaluating as part of first-line therapy. To be eligible for this study, the patients need to meet at least 2 high-risk criteria. First, they have to have a histological diagnosis of double hit or triple hit lymphoma, or if they have a histological diagnosis of large B-cell lymphoma, they have to have an IPI score of 3 or greater. In addition, they have to have a positive interim PET scan defined as a dual score of 4 or 5 after 2 cycles of standard frontline chemotherapy such as R-CHOP or R-EPOCH chemotherapy. So if they met these 2 high-risk criteria, both a high-risk criteria based on histology and IPI, as well as a dynamic risk assessment, the patient could enroll on this study. So following enrollment, they underwent leukapheresis, and once the product has been manufactured, they received standard conditioning chemotherapy with cyclophosphamide and fludarabine, followed by 1 single infusion of axi-cel at a dose of 2 million CAR-positive cells per kilogram body weight. The primary endpoint was complete response rate. At the time of the interim analysis for this abstract presentation, as of the data cutoff of August 25th, 2020, 32 patients received conditioning chemotherapy as well as axi-cel infusion. We found that after a median follow-up of 9.3 months, the best overall response rate was 85%, and the complete response rate was 74%, and 70% of the patients have ongoing remission. The median duration of response, the median PFS, and the median overall survival have not been reached to date. The therapy itself was quite well tolerated. As expected, all patients developed cytokine release syndrome, but most of these were grade 1 and grade 2, with 9% of the patients having grade 3 or higher CRS. And the second most common axi-cel related adverse event was neurological toxicity, with observed in 69% of the patients. But again, most of these were grade 1 and grade 2, with 25% having grade 3 or higher neurotoxicity. All of these were reversible. There were no grade 5 CRS or neurological events. We also observed that the CAR T products in the ZUMA-12 study had a higher frequency of naive phenotype of CAR T cells defined by CD45 or a positive CCR7 positive T cells, which associated with a greater expansion of the CAR T cells compared to the ZUMA-1 study. So in summary, this is the first study to evaluate CAR T-cell therapy as part of first-line therapy in patients with high-risk large B-cell lymphoma. We found that the therapy is quite well tolerated and was associated with a very high efficacy, with a high overall response rate of 85% and a complete response rate of 74%, and 70% of the responses were ongoing after a median follow-up of 9 months. In addition, we also found that the CAR T products have a what appears to be an improved T-cell fitness compared to CAR T product generated after 2 or more lines of systemic therapy, suggesting that moving CAR T-cell therapy to earlier lines of therapy may be beneficial and improve the efficacy of this approach.